Transfer RNA, or tRNA, is a member of a nucleic acid family called ribonucleic acids. RNA molecules are comprised of nucleotides, which are small building blocks for both RNA and DNA. tRNA has a very specific purpose: to bring protein subunits, known as amino acids, to the ribosome, where proteins are constructed. One of the discoverers of DNA, Francis Crick, first suggested the existence of tRNA. At the time, scientists knew that genetic information was kept in the nucleus as DNA, and that DNA carried the instructions on how to make proteins. DNA doesn't leave the nucleus, though, so our cells make a copy of the DNA called messenger RNA, or mRNA. mRNA leaves the nucleus and is bound by ribosomes, the molecular machines that act as the factory that makes proteins. Scientists understood that while DNA and RNA have almost the same alphabet, proteins are very different. Francis Crick proposed that there must be a small molecule capable of translating mRNA into proteins. Other scientists proved his theory with the discovery of tRNA. The job of tRNA is to read the message of nucleic acids, or nucleotides, and translate it into proteins or amino acids. The process of making a protein from an mRNA template is called translation. How does tRNA read the mRNA? It reads the mRNA in 3 nucleotide sequences called codons. Each individual codon corresponds to an amino acid. There are 4 nucleotides in mRNA. If you do the math to figure out how many codons exist, you arrive at 64, or 4 cubed. There is 1 tRNA molecule for each and every codon. Interestingly, there are only 21 amino acids. This brings up the idea that our genetic code is redundant. That is, we have 64 codons, but only 21 amino acids. How do we resolve this? More than 1 codon can specify for an amino acid. This table shows all of the combinations of nucleic acids, or codons, as well as which amino acid is specified by which codon. As you can see, not every amino acid has 4 codons. In fact, methionine only has 1. Notice, however, that each codon has only 1 corresponding amino acid. Thus, we say that the genetic code is redundant, but not ambiguous. For example, the codons GUU, GUC, GUA, and GUG all code for valine, redundancy, and none of them specify any other amino acid. No ambiguity. So we now know that the job of tRNA is to bring an amino acid to the ribosome. We also know that each codon has its own tRNA, and that each tRNA has its own amino acid attached to it. Further, we know that the job of tRNA is to transport amino acids to the ribosome for protein production. The tRNA doesn't become part of the protein, which suggests that tRNA can either be attached to an amino acid or free. We call this charged or uncharged. How does this work? Briefly, an uncharged amino acid goes to a pool of amino acids. Here it finds the one specific amino acid to which it can attach and binds it, or becomes charged. Then it carries the amino acid back to the ribosome, where the amino acid is transferred from the tRNA to the growing protein. At this point, it's important to remember that amino acids are added to the protein in a specific order. The order is contained in the code of mRNA inside the ribosome. How does the tRNA know if it's carrying the right amino acid? That is, the amino acid that needs to be added next according to the codon sequence of the mRNA. Easy. tRNA have anticodons as part of their sequence. An anticodon is the reverse sequence of a codon. In this, imagine you can see a black region at the bottom of a blue loop. This is the anticodon sequence. The anticodon sequence also tells the tRNA which amino acid it can attach to at the pool. The amino acid is attached to the yellow tip of the tRNA. Anticodon sequences work just like codon sequences. Thus, each amino acid has more than 1 anticodon that recognizes it, but each anticodon only recognizes 1 amino acid. In eukaryotic cells, tRNA are made by a special protein that reads the DNA code and makes an RNA copy, or pre-tRNA. This process is called transcription, and for making tRNA, it's done by RNA polymerase III. Pre-tRNA are processed once they leave the nucleus. First, some nucleotides are cut off, and then the remaining 73 to 94 nucleotide long string is folded into the shape you see in this image. Transfer RNA, or tRNA, is a specialized RNA molecule that brings amino acids to the ribosome for protein production. In eukaryotic cells, tRNA is made by a special protein that reads the DNA sequence and makes an RNA copy, or pre-tRNA. Pre-tRNA is processed once it leaves the nucleus. tRNA's role involves multiple steps. First, it reads messenger RNA, or mRNA, in 3 nucleotide sequences called codons. Using this information, the tRNA finds the one specific amino acid to which it can attach and binds it, carrying the amino acid back to the ribosome. The amino acid is then transferred from the tRNA to the growing protein, and tRNA's job is finally complete.